Computer, share screen. We're going to go with 17 first. I thought we would made it to the end. Right here. Okay, so we kind of let's guess we'll start here. Okay. All right, so we can catch up. In my mind, I got to catch up a little bit, so we're back in just a couple of slides. So, methicillin resistant. I think we hopefully you have the understanding there. It's really oxacillin is the antibiotic that we see on our panels. And great thing today is we got panels to do. I think I didn't look at the cultures, but hopefully it's growing. We have some wound cultures and urine cultures. So if you have a positive, then we're going to go ahead and set it up on the combo plate for positive. If you have a negative, it'll just be set up on the MIC. We don't have the combo. Remember, we got the MICs and they wouldn't take them back. So we're just going to use those as our MICs. So the challenge for the gram-negative organism student would be to make your best guesstimate or best guess on what you think it is. Okay, so we may have some play that you can play with some things. If you think it's E. coli, if you think it's cleb and that type deal, we won't have a definitive on our combo, but I want you to see a good MIC with a real life organism. As the organisms we pull out of the freezer downstairs are not going to show resistance. Okay, they haven't been exposed to antimicrobials like a patient's would. So that's the key to today's lab. We'll wrap up what we have going on. Through this week, we'll do the MICs. We'll read those on Thursday. And then it's next week, the week before Thanksgiving. Okay. I've got a blood culture day that I want to do with you in micro. So we want to do that. So that probably might be one of the days next week with looking at the yeast growing. So we'll just grow up everything, start next week, and maybe on Thursday get a look at everything as we start to introduce that after the test. So test for Tuesday or Thursday? Tuesday. Okay, so Thursday we'll start off on our mycology, send you off for Thanksgiving to eat some good stuff, and then come back online, and then we'll start the mycology lectures after that. So I think we're good schedule wise. No, y'all don't want to be here on Tuesdays and Thursdays. It sounds like you're going to be asked to be here on Monday and Wednesday though. So you guys can tell me how you want to go about that tomorrow in immuno class, but think about it till tomorrow. I don't know if you're going. It's one of those things like, well, you know, they could not be here, but then if they're going to be here, do we need to? I don't know. We'll talk about it more tomorrow for immunology. And the ones in the back, I don't even know what we're talking about, but Tuesdays and Thursdays, we won't expect you to be here. Is that a good deal? Okay. So methicillin resistant or oxacillin resistant Staph aureus, MRSA, is a significant problem in the hospital setting because that's where it began. You know, this is a nosocomial infection. Like we've said over and over this semester, this is the operating room infection. This is the patient that came in with not having staph and ended up with staph. So hospital-acquired strains are usually multi-resistant, whereas the community-acquired strain is not. But now we're seeing MRSA just everywhere. So chances are if I got it in the hospital, it's probably going to be MRSA. But if I got it at, you know, just out and about, a staph infection, it's probably going to be MRSA, but now we're starting to see more and more of it. So because a lot of us harbor it and a lot of us carry it around with us and go out, and we infect things. Methicillin resistant, the strains of Staph aureus which carry the MEK-A gene, so that's key for your memory, is to know it's the MEK-A gene that encodes an alternate penicillin binding protein, PBP2A, whose affinity for beta-lactam antibiotics is greatly reduced. So it's a defense mechanism against penicillin, okay, the beta-lactams. At therapeutic levels of methicillin, that would inhibit the transpeptational cross-linking activities of the penicillin binding protein, okay, because they do the cross-linking of the cell wall. The PB2A remains active, ensuring the cross-linking of the glycan chains and peptidoglycan. The limited degree of cross-linking is enough to ensure survival of the cell. Okay, so that's the story of the MEK-A gene in the Staph aureus, giving it methicillin resistant. And this is where we pick back up, is the extended spectrum beta lactamases, the ESBL. I had an ESBL this weekend. It had the ability to destroy carboxypenicillins, tigercillin and carbenicillin, uridiopenicillins, piperacillin, meslocillin, azotreonams, as well as cephalosporins and other cephoxidants, right? So we got an extended spectrum beta lactamases, meaning that organism is now producing beta lactamase enzymes that are really causing havoc with the antibiotics that we're giving. They tend to be resistant to also fluoroquinolones, aminoglycosides. They're usually susceptible in carbapenems, imipenems, meropenems, and doripenems, and ertapenems. The cephalomycins, cefoxidin, and the beta-lactamase inhibitors, clavulanic acid, tazobactam, and sublactams. Got a lot going on in this slide, right? So much. And when we finish this slideshow, we're gonna go to drug.com, and I'm gonna show you a good tool for study, how to study through these names and how to pick out and take up the challenge of knowing the names of the antibiotics versus just the fluoroquinolones being listed, the aminoglycosides being listed. Do you know the names, right? What are the names in those? And that's what we spent all this time on this chapter for. So I can't remember which one I had. It was either an E. coli or Kleb. They're also known for their ability to produce the extended spectrum beta lactamases, but they can also be found on other enterobacteriaceae. Any organisms known to produce extended spectrum beta lactamases requires all penicillins. This is key. This is the note you write up in micro lab and put it on the board until you get this down. If you got an ESBL, requires you need to check your MIC and make sure that the computer will do this, but if you're doing this manually, you need to do this. You need to make sure that the penicillin, the cephalosporin, and the treonams are reported as resistant, regardless of what they look like. Okay, if they look susceptible and they're not growing anything in those wells, if you come up with an extended spectrum beta lactamases, you have to switch all those to resistant. It's just what you have to do. Does that make sense? Because it's not going to be long before they are. So that's why we do it. Traditionally, clinical microbiologists have been taught to think of the enterobacteriaceae in two groups with different resistance patterns. We have the spice. That's easy to remember, right? Got Spice Girls here with Serratia Providentia indole positive Proteus species. Yay, we get a Proteus, make sure it's indole positive. Citrobacter and Enterobacter species. That's the spice. Serratia, Providentia, Proteus, indole positive, Citrobacter, and Enterobacter, all Enterobacteriaceae. And then the Kep bugs, Kleb pneumonia, E. coli, and occasionally Proteus. These two groups differ in their resistant genes that they typically carry. The spice carrying an inducible chromosomal beta-lactamase gene known as AMP-Cs. So you put the spice with Cs. The Kep bugs, on the other hand, carry an extended spectrum beta-lactamase, ESBL genes found on the plasmids. Many laboratories attempt to guide the empiric therapy of these bugs with the use of reporting comments. The Kep bugs now include AMP-C carrying Klebsiella aerogenes. Can y'all make that out? Can you keep, you know what spice? You think you can make that list and Kep? Hope so. So then we get some taxonomical changes with the Kep bugs. Now include AMP-C carrying Kleb aerogenes, formerly Enterobacter aerogenes. Laboratories will need to re-educate clinicians about the split of the Klebsiella genus, which now contains organisms carrying ESBLs and those carrying AMP-Cs. Furthermore, there are numerous guidelines of CLSI. Y'all know what CLSI is? I don't think I've ever got to ask you that, or have I? Clinical Laboratory Standards Institute. Y'all ever heard of that group? They're kind of the group that puts all the procedural stuff together. Publishes little blue books that you'll find in your lab. And it might be as simple as the phlebotomy procedure, right? And it'll give what CLSI recommends for phlebotomy. Like the procedure, tell you what to do. Okay, so we will lean on CLSI for our blood culture procedure that we're going to do next week, because there is some controversy with blood cultures versus real versus recommended. The perception, we've always done it this way. Why are you doing it this way? Shouldn't you always do it this way? I'm ordering it this way. You should do it this way. And we're going to talk about CLSI's, what they recommend, because that's the gold standard for everything that we do is CLSI. They put it together, they update it, they modernize it. They publish a few every year. They'll publish a new procedure, just really redoing the old procedures, adding and subtracting. So it's really neat to have. It's kind of the, the go-to when you got to prove somebody different. And we'll talk more about that with blood cultures in lab. So they're saying for Enterobacter or the Kleb genus that will now need major revisions. ESBL testing has been applicable in the past for all Kleb, but not applicable for the new Kleb aerogenes, which was also known as Enterobacter. All right, a lot of stuff in that last little bit, right? Okay, so we're gonna click off that one. I'll stop the share. And what I want to do is we want to pull up this website here, and we'll share this once I get it up. Drug.com, bacterial infections. Okay, list of medications and compounds. This is really great. I love this thing. Just look at all this. So when we do our MICs on Thursday, we've got all these, most of these listed on our MIC. But what you'll see is a lot of names you may have heard, maybe not have heard. But it's definitely a guide. So it's just drugs.com, bacterial infection, listed as drugs used to treat bacterial infections, medication list. So this is really simple. So I love this. So you click on amoxicillin. It'll tell you the generic name, amoxicillin. It'll say the brand names are Amoxil, right? So we're challenged. Remember I told you to challenge yourself to start putting that together. It is an aminopenicillin drug class. So I'll click through a few of these. Oh, I was supposed to share my screen. Why not? Zoom has no idea what I'm doing. Sorry, Zoom. Is it here? Yeah. They're supposed to speak up or the volume's up, so yeah, spoke up. All right. Clindamycin, Cleocin is another name, goes under. Drug class, lincomycin derivatives. Ciprofloxacin is a quinolone. Ciprofloxacin, always also known as Cipro. Oh, this is fun, right? And you can go do some, you see these other things. If you're pregnant, central, what is CSA? I don't know all these things yet. So. Doxycycline. It's tetracycline. Goes under doxy. We talked about doxy with what infections? Rickettsial, right? Tick, tick. Okay, yeah, all right. Let's see. Levofloxacin, Levaquin. We went over that, right? Quinolone. Amoxil. We already covered that, right? So it kind of goes back and forth. Augmentin is amoxicillin with clavulanate. So it's a combination of the two. Beta-lactamase inhibitor. Augmentin is pretty fragile. And we talked about that. The azithromycin dose pack. The macrolide. There's Cipro again. There's amoxicillin clavulanate. Keyflex, that used to be one I got all the time. Cephalexin, first generation cephalosporin. Bactrim, sulfonamide, sulfamethoxazole trimethoprim. Levaquin again, Bactrim. We already hit that one. Sulfamethazole and trimethoprim, Bactrim. Zithromax, macrolide, azithromycin, Zysystemax, so azithromycin, Z-Pack, Zithromax, penicillin, vancomycin is vancomycin. Okay, so it's a good way to play with the names, most common names that we'll see. And it kind of gets you where? It kind of gets you hopefully on the same page as your nursing staff. When they throw the other names at you, you kind of have it grouped up. You kind of know what's going on with the names. All right, so let's pick up our new PowerPoint. We'll stop that share, start another share. We got chapter, if I open that other PowerPoint, I don't think I have, here it is, 18. And that's where we are today with this lecture, right? Not near as long as we've been hanging in here. And this is what, oops, I wanted to share first. Hang in there, Zoom. Share my screen. Where'd it go? Is it sharing? Do you see susceptibility part 2? All right, good. I think this is chapter 11. I do know, so it's something we're going to be doing on Thursday for sure. So we're going to work our way through what is susceptibility. So we kind of set up, part 1 with all the different classes and the mechanisms behind how they work, what they're going to work on, whether they're a DNA inhibitor or a transcription inhibitor, or they're cell wall inhibitors. So now we're going to talk about susceptibility testing. So what we do is an effort, determine the effort a microbial agents have on microorganisms, so the appropriate therapeutic agent is prescribed. So that's our number 1 role. You can say, oh, I just love that I can tell the doctor there's E. coli in the urine. That's great, right? Doctor wants to know, what therapeutic agent I need to have them on. They're not really worried that you got E. coli or you got Staph aureus. They want to know what end result, what's going to work, what's going to get rid of it. They're all about what results. Okay. So measuring that response that the microorganism has on various inhibitory compounds is important because how a microbe responds is antimicrobial agent cannot be predicted, i.e. susceptibility of staph to penicillin. Okay. Definitions. We have this MIC. Maybe you've heard me mention that. Maybe you don't know what I was meaning when I said we're going to do MICs today. MIC is the minimal inhibitory concentration, which is the lowest concentration of an antimicrobial agent that visibly inhibits the growth. Okay, so that's what we're dealing with MICs. Minimum bactericidal concentration, MBC. You're not going to hear MBC very often, if anytime. That's the lowest concentration of an antimicrobial agent that results in the death of an organism. All right? So minimum inhibitory concentration is the one we're looking for when we do these MICs on the bottom of our panels of our microscan panel to see what we've got. So we have some definitions of the effects of the antimicrobials. So we have the first one, which is synergistic. Greater antimicrobial activity from a combination of agents than either acting alone. So what was the one I just popped up on drugs.com? What was one of those that had 2 in it? Remember? Y'all paying attention, flipping through. It had a name. What was the name of that one? Anybody remember? Zoom, you remember? Y'all were taking screenshots. What do you remember? I'm just kidding on the screenshots. What do y'all remember from that list? Were y'all even looking at the list? You were? Sulfonyloxazole and somebody trimethoprim. What is the name of that one? Nobody remembers? Okay, stop the share. Go back. Here we go. I got them open. I remember. Which one had 2? Levaquin, amoxil.
Can you zoom in on it? I mean, Augmentin has one.
Augmentin? That has 2, right? Need to zoom. Somebody wants to. I don't.
I heard like the blue ones.
Is that what you want there?
Yeah.
How's that?
Yeah.
Okay. Augmentin, right? Amoxicillin and clavulvinate, right? Yeah. Okay. So yes, that would be an example of one. Okay, good. Now let's go back to sharing. Or did we ever quit sharing? Did I quit sharing? Quit sharing. Goodness. Okay. Share the screen. Go back to PowerPoint 18. Let's go from here, from the current slide. All right. So synergistic, greater activity when you have a combination than either one acting alone. Antagonistic effect, the combination of antimicrobial agents, is less effective than either used alone. And then indifferent, the combination of agents neither more nor less effective than each other agent used alone. So where might we get an accidental antagonistic effect where a combination is less effective than either used alone? How could you see that happening with a patient? Has anybody ever taken an antimicrobial agent? Let's start there. Anybody? Who's all had an antibiotic in their life? Remember taking it? Some that never have taken it? Yeah. How could an accidental antagonistic effect occur? When you self-prescribe yourself. When you find it in the shelf or in the cabinet and you go, I think I have an infection. I think I'll pop a few of these. And then you end up going to the doctor. The doctor goes, well, I'm going to prescribe you a different one, right? I'm going to prescribe you this one. You didn't tell him you had taken some others at home. And then all of a sudden you get an antagonistic effect because you took one that was for something else. You didn't know what they were used for. You just said it was maybe not even antibiotic. Who knows, right? Do y'all do that in your medicine cabinet? Or do you label things? Do you put little things on the tops that say pain killer or antimicrobial or?
Well, I take all my antibiotics for one.
I've
never had leftover antibiotics.
Okay.
My sister has though.
Good. I'm glad you brought that up. So here is one of those moments. Okay. I know the doctors are gonna. No, the doctor shouldn't mind. Shouldn't mind this at all. Okay. So why did you take them all?
Because I don't want to become resistant.
You don't want to become resistant. So there's not an imaginary number. There's a standard number. What is the standard number of taking antibiotics? How many number of days?
Like 5.
5, 10, all right?
Yeah, depends on the dose though, doesn't it?
Depends on the dose. It's all relative. Okay, so y'all listening? Let's see all your eyes up this way. Okay, this is like the greatest news you ever hear. Y'all know where those days came from? So we kind of got this. We kind of got the scoop one time in a presentation from an ER doctor. The scoop is that the drug reps want you to believe in their drug, their antibiotic. So they wanted to extend the dosing days to make sure that it clears the infection. So the drug reps came up with this, let's go, the recommended dose, doc, will be 10 days. Because we sure don't want resistance to show. Do y'all know on average how long it takes an antibiotic to clear a bacterial infection?
Like
2 to 3 days. Clear it. Not linger. Not, oh, it's got to give 5 more days of treatment because it's 2 to 3 days. And it's gone. Okay? So, doctors are still prescribing it because that's the recommended prescription time. But that was the scoop that we were informed of many years ago, that the antibiotic itself is so good, all right, that it will take care of things in a couple of days. Gone. So what happens when you do 10 days? What are you doing?
You're just making sure that everything's gone.
But you're also wiping your normal flora, right? So when you wipe that normal flora away, like you would in a hospital, C. diff arises. C. diff is a major problem right now because we think the 10-day dose. Okay, keep that in mind. So that's kind of like ta-da, big moment. Big moment when we're talking about microbial susceptibility. But yes, there is that fear of if I don't take them all, then that E. coli is going to what? Mutate. The leftover E. coli is going to figure out a way to change and mutate to become resistant, right? So that is the fear. So that being said, so how did we?
Pack it for 3 days.
Huh?
Pack it for 3 days.
Sure, I'm good with that. Then you got some left in the cabinet for the next time you get it.
I only say that because my sister has made drugs resistant to her by taking it for 1 day and feeling better. And
Yeah, don't just take one single couple of doses. Yeah, it needs a few days. Don't, but like, what is the new? Who said 5?
I said fine.
So who's the one that does 5 days? Z-Pack. Is Z-Pack new? I mean, it's compared to penicillin.
Oh, yeah.
Yeah, I mean, it's come around. It hadn't been around forever, but and that's where you get, oh, well, Z-Pack must have a bigger dose because you only have to take Z-Pack 5 days. Maybe Z-Pack maker goes, let's just give them 5 days, and that'll assure us that we made it past the 2-3 days, but we don't have to go 10 days. They still charge like you're going out 10 days, don't they? Z-Packs aren't cheap, I don't think. But I don't know. I hadn't had one, so I've seen them. Okay. All right. Any other questions? That's good.
What is CIDIC short for?
Clostridioides difficile. We've already had that one. Yeah, it used to be known as Clostridium. We changed that name. Clostridioides difficile. Hospital-acquired infection due to antimicrobial usage, overusage. Patient loses normal flora. Clostridioides difficile finds a home, starts producing its toxin, irritates the lining of the bowel. The bowel starts to fill up with water, and you start having diarrhea. It's usually a diarrheal episode after antimicrobial treatment. You went 10 days. But most of us, right? Because the doctors fear that you're going to feel better after a few doses and say, I'm done. It's okay. Just give it a few days and be okay. But yeah, I have a hard time convincing my wife to quit taking them. Like, hey, aren't you feeling better? Hey, how many days have you been on that? I think you're good. All right. I think that UTI is probably taken care of, but we check it. Come back and check your urine again, and we'll make sure it's okay. But you shouldn't see it. Okay. So we have bacterial agar diffusion. DAD, disc agar diffusion. So this is old school. We can still do this. We take a Mueller-Hinton agar, and we can grow an organism across it, streak it across, and then lay down a disc of antibiotics. All right, so that's what we're doing. So the bacterial inoculum is applied to the agar plate. Thin paper disc, kind of like our A disc, our Taxo A, and our P disc. Y'all remember those? Did we use those for strep? Strep pyogenes. Remember way back when? I know it seems like 2 years ago when we did that, but it was just a few weeks, a couple months ago. So the thin discs are impregnated with the antimicrobial agent. You place that on it, just like you do the A and the P disc for strep. And the plates are incubated. The plates are examined for a zone of inhibition. So what a zone inhibition is where the concentration gradient of the antimicrobial, right, will start to form a zone of clearing. As it diffuses in the agar, the organism will not be able to grow around that disc if it's sensitive to it. At some point where there is an amount of antimicrobial agents not sufficient to inhibit the growth, that's where the zone stops. So this is the zone of inhibition surrounding the disc. The point at which this occurs is related to the MIC. MIC is what?
Minimal inhibitory
Concentration, exactly. The disc agar diffusion test has only been standardized for rapidly growing aerobic facultative bacteria such as staph, strep, and enterobacteriaceae. So our main ones, our very rarely do you fall out of that group, right? Staph, strep, and the enterobacs. It's not applicable for the slow growing or anaerobic bacteria. So the disc agar diffusion test provides qualitative results only and is reported as susceptible, moderately susceptible, or intermediate or resistant. And we said hold off on the intermediate, right? We said that's just an unknown area if you find that. And if the proper method is not carefully followed, variations in the zones will occur and the test is neither accurate nor reproducible. So we have some Mueller-Hinton downstairs. Got to use it, all right? So we're gonna bring this out with some disc. We've got some organisms growing down there, so we're gonna re-streak Mueller-Hinton today. And then we're gonna lay the disc. I don't know if I have them all set up in the little dropper. You might just have to manually place them on there, but I just want to let you see on Thursday. It is Tuesday, right? Thursday, if we can we create some zones of inhibition. And hopefully that'll match up with what? Your microscan plates that you're doing for Thursday too. So it's good that we have some Mueller-Hinton downstairs. 4 or 5 wells isolated, 4 or 5 well isolated colonies. Sorry, this is the test method. Free from contamination. So that you want to find a single growth, hopefully. This because you don't want to mix 2 organisms onto a Mueller-Hinton for this. So you don't want to have to Same thing you would do with MICs with the microscan. Sorry, I refer to it as microscan now. We don't want to place 2 different organisms against 1 antibiotic. Might not work out too well. Colonies are placed into a 1 to 2 ml Mueller-Hinton broth or a TS broth and incubated at 35 degrees to reach a log phase of maximum. So what we have is actually dividing cells on plates. We got blood and chocolate downstairs in McConkey. After incubation, the suspension is adjusted to match a 0.5 McFarland, which is a 1 to 10 colony forming units per mil, and you may have to dilute it. So we're going to kind of skip that step because we're not going to measure these. We're going, well, I say we can make that up. I think we can make that dilution. I think I've got the stuff. So I think we can do this. Inoculate the Mueller-Hinton agar plate using a sterile non-toxic cotton tip swab dipped into the broth. Excess inoculum is removed by rotating the swab against the tube, 3 different planes over the entire swab plate. Wait 3 to 5 minutes for absorption of the broth, but no longer than 15. Generation time is 20 minutes. And then apply the antibiotic disc. Gently press those discs into the agar, ensure contact with the agar, free diffusion, invert the plate, incubate for another 18 hours in a non-CO2 incubator. So interpretation is we look for the diameter of the zone of inhibition measured using a ruler or a caliper. And then, you ever seen a caliper? No. It's a little ruler that has a little, I like to call them teeth, right? It starts like this, and the ruler's my arm, and then it like this, it sits out so you could actually go from one point to the other. Then it tells you how far that one point to the other is without actually putting it on top or pulling it over. So they're pretty neat to use. I think we have some. I think we at least have a few. And then what we're doing is the point at which this occurs is related to the MIC, antimicrobial agent of the organisms. Plates enriched with blood can be used for fastidious organisms, but may be read from the top of the plate. Quality control strains of E. coli, Staph aureus, and Pseudomonas should be tested at intervals to which new lots of Mueller-Hinton agar plates. That all being said, this is what it looks like. All right, so you can see the disc diffusion method. So we would measure the zone, right, from one side of the zone to the other, right, through the inhibition. So this would be an example of what? No zone, right? Pointing to the one with no zone. There's a couple on here with no zone of clearance. Somewhat clearing over there to the left, but not near as nice. And then got one here that would be total resistance here. That might be the intermediate, you'd say. But usually it's the distance that you measure, but you can kind of tell these are all sensitive. This organism is sensitive to several of these, most of these. Kind of resistant to 1 or 2. So again, closer up. So you have these at different concentrations. These are different antibiotics. It gives you an idea. So what factors influence the zone sizes? There's two. Critical concentration of the antibiotic and the organism reaching a critical mass. Concentration of the antibiotic is affected by the rate of diffusion. The critical density of the organism is affected by the rate of growth of the organism. So what you're really saying is, I sure am glad we have MIC panels to do this with, but it's going to be fun. So that inoculum density is the most important single variable. Like, how much did you put in there? All right. The zone of inhibition is caused by a critical concentration of the antibiotic, which has the ability to inhibit the critical mass. And this is key, this is great test questions. You go ahead and put a star, circle, whatever you need to do, because I'll ask you to distinguish these 2 or 3 things, 4 things. If the inoculum is too light, hey, I didn't get enough colony in there. So I didn't get enough. The critical density of the organism takes longer to achieve. The antibiotic has more time to diffuse and have an effect before the critical mass is attained, resulting in a larger zone of inhibition, falsely susceptible. Okay? So if you don't put enough organism on there, the antibiotic has a larger zone, so a false positive for susceptibility. The zone is too big because you didn't put enough inoculum on there. If you put too much inoculum, the critical density is reached far below the antibiotic, right? Diffuses, or a chance to have to affect the growth. So there'd be a false resistance, so a smaller zone of inhibition. So this is very important because we started by talking about picking colonies, picking a single colony with your prompt has a very small point to it, just a very small opening, and then you have a little, the little grater or whatever you want, a cleaner that you pull off through the stylet to clean it even better, because we don't want too much. Okay, so stab a colony, get it in the point, the tip, and that's it. So remember how we started? So we watched y'all make those McFarlands. Y'all made like 4 and 5 McFarland, right? 3 or 4, not 5. 3 and 4 McFarlands, and y'all were like wiping every colony off the face of the plate to get there, right? So now it's not quite that difficult. But that would be a large inoculum. Agar concentration. 1.5 to 2% is recommended. This concentration allows free diffusion of the drug into the medium. Temperature. Zone sizes increase as the temperature increases due to increased viscosity of the agar, which leads to increased diffusion. Lower temperatures prolong the growth, so it takes longer to reach critical mass, so you get larger zone sizes, low temps. Do not stack the plates or center plates take longer to reach the proper temperature. So one of the things that Beckman Coulter recommends for those microscan panels is don't go any higher than 5. All right? Don't go any higher than 5. pH. pH can affect the activity of certain antibiotics, such as penicillin. Penicillin is affected by a low pH. The growth medium must be free of fermentable carbohydrates. Production of metabolic acids. That's why we use Mueller-Hinton. Plates cannot be incubated under increased CO2. Cannot use CO2 incubators because carbonic acid is formed on the surface of the agar, creating a decrease in the pH. Uniform depth. millimeters. When plates are too thin, less of the antibiotic diffuses downward and more diffuses horizontally, leading to increased zone sizes. So we got 2 students down there pouring plates this afternoon. You know, it's not a difficult task, but you know, basically when you're pouring plates, you take the diameter, you take the plate, you pour the agar hot in there until it covers itself, right? And that's usually enough. So it's not an exact science to get 4 millimeters in there, but works pretty well as long as you're pouring, as long as you're doing it the same way, you get a uniformity in depth. But you've seen some of the plates. You saw, I think, what was the one chrome agar plate about evaporated back. You know, it was losing its ability to stay out there. That's why they expired. Disc storage, we want to make sure they're stored in the fridge. We warm them to room temperature before opening to reduce condensation on the disc and watch expiration dates. So as I'm talking through this, when I'm talking through all this, should we be making notes for strep, streaking with bacitracin, the A disc? Should we be making notes when we streak for strep pneumo? We use the P disc. Should I ask what the P disc has in it? I gave you the A disc. What's the P disc have? Ooh. Okay. We should be. The things that we're talking now, the Mueller-Hinton disc diffusion, we should do that with our strep. Same thing. How many of us take the Toxo A right out of the fridge and put it down on there and go? Right? Don't have time. Could be a factor influencing the zone. Organisms which are methicillin or oxacillin resistant should be considered resistant to cephalosporins, methicillin resistant Staph aureus. Use of oxacillin discs to predict susceptibility to strep pneumo to penicillin, the DAD test does not accurately detect penicillin resistant using the penicillin disc. All right, we're going to pick up a little bit because I know y'all are about to, minds are getting a little tired. We have a lot. This is in, again, chapter 11. Dilution procedures. Y'all love dilutions, right? Your favorite thing to do in immunology this year was dilutions. You right? I know it was. I learned so much. Dilution procedures. Susceptibility tests include broth dilution, agar dilution, and microtube broth dilution. These tests are quantitative because MIC values are obtained and qualitative information may be reported as susceptible, intermediate, or resistant. Remember, I've warned you, I think, enough on intermediate. You should be conscious of that. Serial dilutions of the agent are made and the organism to be tested is added to different concentration of the drug. Interpretation. Dilutions are examined for growth through turbidity. MIC, the concentration of the first tube where there is no visible growth. So instead of putting it on an agar plate, we're doing a tube dilution, serial dilution. The MIC is where there is no visible growth in that tube. So serial dilution down, come up with the value. These methods are good for slow growers and anaerobes, new antibiotics, antibiotics such as polymyxins, which diffuse poorly in the agar. For systemic diseases, effective therapy requires levels 2 to 4 times the MIC because some of the drug is metabolized by the liver and excreted by the urine once it's given to the patient. These actions would tend to decrease the availability of the antibiotic in the serum, cerebrospinal fluid, tissues, wounds, and abscesses and tissue fluids where infections occur. For urinary tract infections, effective therapy requires levels 10 times the MIC for the antibiotic to be effective because it's excreted into the urine by the kidneys. So keep that in mind, that we have an MIC, the minimum inhibitory concentration, but we got to increase that level. We can't just go with the MIC minimum. Urinary tract, we go 10 times higher. So here is our turbidity test. All right, MIC determination. Looks like we're going in reverse, right? 0.25 on the right. We increase the concentration of the antibiotic. And 8 is the level as to which this is the MIC, right? So if this is a urinary tract infection, how much antibiotic would we want to go with? 80 micrograms, right? Everybody see that? So this is the turbidity growth that leaves us at the MIC. Staph aureus doing the same, looking at a different way of doing with the tubes. Cloudy tubes indicate growth. Here's what we're going to be looking at more on Thursday. MIC interpretation as we went, right? The higher concentration here, get out to there where the first one shows up. Point at which growth is inhibited is the minimum inhibitory concentration. Yeah, is that confusing to anybody? That looks weird to me. Is anybody having trouble with this one? Everybody good? Nobody's confused? Higher concentration here, inhibiting the growth. All right, sensitive, 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 resistant. We're diluting, right? Not going up in concentration of dose, diluting the original dose. I didn't want y'all to be confused. All right, serum bactericidal test. Schlichter test. The test measures effectiveness of an antibiotic in vivo. The lowest dilution of a patient's serum which kills a standard amount of bacteria. Method of the Schlichter test is a serial two-fold dilution of serum from the patient receiving antibiotic. Standard amount of bacteria isolated from the patient is added to each tube and incubated. Observe all tubes for growth. Using a 0.01 mil calibrated inoculating loop. How many microliters is that? Ms. Bednar was just bragging about her lab math down there today for principles. What is 0.01 mils? How many microliters? Should be automatic. How many microliters is that?
One.
Huh?
One.
1? Nope.
10.
Huh?
10.
10. 10's right. But thanks for, yeah. Don't y'all seem like you're so timid to say, just say, I'm not gonna take points. After these plates are incubated for 18 to 24 hours, their growth is observed. First dilution showing growth of fewer than 10 colonies, taking as the endpoint. Ooh, that was easy. Assays of antimicrobials. Why is there a need to determine antibiotic concentration in body fluids? Why would we need to do that? This is our clinical chemistry. Gentamicin pops up, right? Because we use toxic antimicrobials. We talked about that last week. So we need to do what? We need to determine the concentration. The use of agents for treating closed space infections such as meningitis when the penetration kinetics of an agent are not predictable. The desire for a more precise therapy evidenced by demand for MICs and MBCs, pharmacokinetics to study the excretion of the degrading of the drug. Investigation in determining drug pharmacokinetics requires data on trough and peak levels before new antibiotics can be approved. So peak and trough, that's what we use it for. The trough is this is where we said we were coming to this. Level of medication before administration of the drug. Samples are drawn 30 minutes before administration of the next dose. Okay, so as you're seeing this laying out for you, I hope you understand. Well, for many, many years, everybody in the lab felt trough was the start of the process. The peak drawn after the dose, okay, sounds just like what I said, is given and time so the anticipated concentration is highest. Depending on the route of administration, IV, IM, or oral, peak levels are observed 30 to 90 minutes after administration. As you see, trough is starting to get put in front of peak, right? I'm going to yield to the pharmacist who said, no, I want the peak and then the trough for the, after a dose to the next dose. Don't do anything before that dose I'm telling you I want info on. All right, agar diffusion method. Again, an overnight broth culture of seed organism is added into a melted Mueller-Hinton agar and mixed. The molten suspension is poured. Standardized solution of graduated concentrations of antibiotics are added to the filter paper disc, and patient serum to be tested is added to another disc. So what are we doing here? We're doing an agar diffusion on the patient's serum to determine how much antibiotic they have in their serum. I don't know if y'all picked that up or not. That's what we're doing. The plate is incubated. The zone sizes around the disc containing the standards are measured and plotted on the graph against the known concentration of the standard. Zone sizes around the disc containing serum are measured and concentration of the interpolated from graph. All right. The good thing is you're going to get to play today and Thursday, and you're going to get to do some of this and see some of this. So hopefully it'll bring all of it around. We have one more lecture right before the exam. So we're just kind of probably going to do a, I think on the syllabus it's called Modern Practices in the Micro Lab. We'll probably stay with micro in this vein. We probably won't switch topics. Modern Practices is kind of like a wrap-up lecture at the very end of the semester. So that's what we'll do Thursday. Exam Tuesday, but we will have lab Tuesday and Thursday next week, just so we can get done and get the yeast and the yeast and the fungi growing next week, so you can see that on Thursday. Wrap that. All right, see you downstairs at 12:30. Gonna end the share. End the Zoom. See you guys later.